In this lecture, we go through the process of glycolysis, and we'll also take a look at the end at the results so that you know what the products are that are produced. So this is our overview slide. This is a good little animation for you to take a look at. If it doesn't play directly out of the PowerPoint slides, hopefully you can copy and paste it directly in. I did check it recently. The link is still working, but at any given point, links can give out on you. So let me know if something doesn't work. So we're going to go through the 10 reactions of glycolysis. These images are posted for you on the Moodle page. They are there also blank for you to quiz yourself. I will put these on the exams. Now your exams are going to be online through the Moodle page on the you'll do them in class on your laptops. However, these images will be paper printouts that I will hand you, and you will be asked to label these images. So you will have an image to look at for glycolysis, these two here. You have an image to look at for the Krebs or ETS or citric acid cycle, and then another image for ETS. You don't get all three of them on the exam, you get one of them on the exam. And again, they will be, it's just easier to do this on a paper copy. So that will not be on the electronic portion of the exam, but it'll still be given the same time the exam is given. So what you'll see, again, it'll be these pictures. It's actually usually I kind of shrink them down so they fit on a single page. I remove a lot of what you see here and leave for you the blue balls. They represent the carbons that are part of your C6H12O6. I'm trying to think if I've got a picture of glucose, or in a previous slide, the glucose molecule C6. I'm going to just jump back if you'll recall back in the earlier lecture. Here's your C6H12O6. This here is your glucose molecule right here. So in this picture, we're looking at a single glucose molecule, and we just are representing in this picture the carbons, the C6. The H12 and O6 are still here. They're just not represented by any specific little balls. So understand that they are there. In fact, the next slide, this is probably a better representation of the glucose molecule. This is what it really looks like. That's a little bit more complex. I have the picture here so you can at least see what it is, but this is the image that is used on the exam. So I remove that it's glucose. In the red boxes here, or in the red writing, are the actual enzyme categories. Those will be gone. I usually do remove the names of each of the molecules. You're not required to know the names of each molecule, but I just usually remove them. I remove the ATP use and formation, the NADHs, the water, they all get removed. The pyruvic acid at the end is all removed. And we're going to walk through this here in a second, but I want you to know I clean this picture up, and all that's really here are the balls and the arrows, and then all the little balls, and then the little pinkish red phosphates stick around. And then you're given certain, I just label whatever enzyme categories here. I'll ask you to write in where is ATP used, where is ATP formed, where is water released, where is NAD reduced. Those are the kinds of questions that I ask. So what's the end product of glycolysis? And so you'd have to tell me pyruvic acid and so forth. So those are the questions that are asked from this image. So as I walk through them and as you learn this, you're going to label it, but then I usually ask a question or two about it as well. So pay attention not just to how it looks so that you can copy it over again, but understand what the process is that's going on here. And so as you walk through this, just keep in mind that category of enzymes. So we start with a full glucose molecule. This is taking place inside the cytoplasm. A lot of professors may tell you this is an anaerobic process. This is important to understand how I want you to know this. The term anaerobic often implies that a process will not occur with oxygen, and that's incorrect with regards to glycolysis. Glycolysis happens independent of oxygen. So what that means is if there's oxygen there, it'll work. If there's no oxygen, it'll still work. So we don't need oxygen one way or the other for this. So to say it's anaerobic implies that there would have to occur in the absence of oxygen molecules, and that's not true. It doesn't need oxygen to be gone. It's not truly an anaerobic process. It just is a process that does not require oxygen. So be careful with that. I usually don't ask any kind of questions that will trick you in that area, but I want you to understand what I'm saying here. We're in the cytoplasm, so glucose has managed to get inside the cell. Hopefully you remember what hormone helps do that. And if you're thinking to yourself, hormone, you should know insulin is the molecule, the hormone that allows glucose to enter into most of our cells. Now, some of our cells, like in the brain and certain areas of the body, glucose can get in without the help of the hormone insulin. But the majority of the cells in our body require insulin. So insulin allows the glucose in. Now we're inside the cell, and there sits the glucose molecule, and all these enzymes are hanging out ready to go to work. And so what are they going to do? They're going to take that glucose molecule and going to say, all right, now I'm going to get some energy from that. I'm going to try and help make some ATP. So in the process, though, the very first thing that's going to happen is we're going to have an enzyme, a kinase, that comes along and attaches a phosphate. Well, that process from going from just the 6 carbons to the 6 carbon, remember that oxygen, hydrogen is still there, but we just don't see them in our picture. But the 6 carbons pick up a phosphate. We now have a glucose 6 phosphate. Well, in order to get that, we needed to spend a little energy. So an ATP comes along, and the energy from that allows this bond right here to form. So we have glucose 6 phosphate. Now you've got an isomerase that rearranges this molecule. Now it looks exactly the same here because you cannot see the oxygens and the hydrogens. But if you could, you would see that this changed its shape. So we went from glucose 6 phosphate to fructose 6 phosphate, a rearrangement. So an isomerase did that for us. Now we're going to pick up another phosphate. So we use another ATP. And where did that ATP come from, you might be asking? Well, that's just available in the cell from another glucose that produced some other energy at some other point. So we have energy in the cell waiting to take action here. Now, if we run out of ATP, if we have nothing to make it, eventually the glucose could come into the cell and just sit there. If there's no ATP, the very first step never occurs. So you have to have energy in order to get this process going. So now we'll look what we've got. When we brought that next phosphate down, we put it on the first carbon. So we had on the first carbon and the 6th carbon. So here we have the 1,6-diphosphate. So the transferase did that for us, the kinase. And now we get a lyase in there. Remember, lyases break the molecule apart. That enzyme splits it. And now we went from this 6-carbon molecule to 2 3-carbon molecules. Now this is the trickiest part of our image. We actually have one molecule, glyceraldehyde 3-phosphate, or GP. And this one, you'll see there's an arrow. It says ready to roll, basically saying ready to roll. It's going to continue on to the top of the next page because it's in the right form. It's got 3 carbons with the phosphate on basically the 3rd carbon. So that one's good to go. Nothing else we have to do with this one. We're ready for the next step. But this one over here, dihydroxyacetone phosphate, or DHAP, that's got its phosphate on the 1st carbon. It's not quite ready. So we bring in an isomerase and we rearrange this one. And then we move that phosphate over here until it looks just like the GP, and now it can go forward. So what's tricky here is that the isomerase only acts on this side, the DHAP. This one continues forward, but this one will too. As soon as it rearranges, it also will continue forward. So we now have 2 molecules, once we've fixed it, going forward. And so you'll see up on the 2nd page here, in front of everything is a 2. No 2's over here. But in front of every one of these is a 2. Those 2's are there to tell us, wait a minute, there wasn't just one molecule that went through this pathway, but 2 molecules that go through the pathway. So although we have just one picture, it's happening twice. So keep that in mind. So just 2 of everything. So the GP comes up here, and now we have this very powerful oxidoreductase. And this oxidoreductase is responsible for an oxidation reduction reaction. NAD is going to be reduced. It's going to pick up a hydrogen. GP is what got oxidized. So we took a hydrogen from here and we gave it to NAD. So a bond broke here, hydrogen came off. It got picked up by this coenzyme, this electron carrier, and now it's got a hold of it. And remember, the 2 is there because each GP will do that. Okay? In the process of this enzyme reaction, again, the 2 is there saying it happened twice. An inorganic phosphate, not one that was from ATP, but just one that's present in the cell, helping you think, wow, I guess phosphate's important in my body. Look at all the things it's doing. It joins on, and it's got this very high energy, quick to break off bond right here. So it joins on, because remember, this picture of 3 carbons with a phosphate that we had here, that didn't change in the oxidoreductase as far as the carbon phosphate group goes. It was still the same. It was a hydrogen that was lost. Remember, C6H12O6. So now we've got this 1,3-diphosphoglyceric acid. And now, with the help of a kinase, we can make ATP. This phosphate will break free and form, well, the ADP that was sitting from over here forms up and we make ATP. So cool. We've done that twice now because, again, we've got 2 GPs, so we've just netted 0. We used one, picked up 2. So we're right at this point in time, we are net 0 on energy. Well, now we've got this molecule left here, the 3-phosphoglyceric acid, because we gave up the other phosphate. Isomerase will rearrange it, and you'll see that phosphate move to the center. Then a lyase is going to come along, and really, this is where this could also be a hydrolase. But really, the actual enzyme enolase that's involved here is technically now categorized as a lyase. It causes this H2O to split off. So, although the carbon didn't split, we got some hydrogens and oxygens out of the C6H12O6, or what's left of it, and we got a couple of water molecules. So the more appropriate category here is lyase. I will take a hydrolase, but if you follow along what's here, you're going to be great using a lyase. So now, in the process of releasing that, this bond is weakening and ready to break free. So a kinase comes along and says, great, I will take this and I will make a couple more ATP. ADP, we're in the cytoplasm from some other use, and we'll regenerate a little more ATP. So that in the end, we're going to net 2. We used 2 here, and we actually made 2, 4 here. So our end product, the 3 carbons and the hydrogens and oxygens that are left, is pyruvic acid. And there you have glycolysis. So what is our single glucose molecule going to give us? Remember, there's 2 because we had 2 GPs going forward, 2 pyruvic acids. We netted 2 ATP. There were 2 NADHs up here. They're holding on to some energy for us. And we got a couple of water molecules. That's your yields of glycolysis.